Hi, y'all. I wanted to do just a quick overview for you to kind of help explain 13.1. I know this first week, that's a lot of reading, but if you'll remember before the break, that's what I told you I was going to lay a lot more reading on you. So let's just think about it. DNA is the genetic material of cells. So the sequence of nucleotide bases in the strands of DNA carry out some sort of code. So in order for that code to work, the cell must be able to understand it. So what exactly do these bases code for? And what is the cell's decoding system? How does RNA differ from DNA? So there are three important differences between RNA and DNA. So the first one is the sugar in RNA is ribose instead of deoxyribose. And number two, RNA is generally single-stranded, not double-stranded. And the third way that RNA differs from DNA is RNA contains uracil in the place of thymine. So on the role of RNA, and sorry, having a little asthma flare-up, so I'm drinking a little coffee. Genes contain coded DNA instructions that tell cells how to build proteins. So the first step in decoding these genetic instructions is to copy part of the base sequence from the DNA into the RNA. So RNA, like DNA, is a nucleic acid that consists of a long chain of nucleotides. And RNA then uses the base sequence copied from DNA to direct the production of proteins. So when you compare RNA and DNA, each nucleotide in both the DNA and the RNA is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. So there are three important differences between the RNA and DNA. Sugar in the RNA is ribose instead of deoxyribose. RNA is generally single-stranded, not double-stranded. Third, RNA contains uracil in place of thymine. So these chemical differences make it easy for the enzymes in the cell to tell DNA and RNA apart. Okay, so next we're going to look at the roles played by DNA and RNA that are similar to the master plans and blueprints used by builders. There's a section on this in your book. So a master plan has all the information needed to construct a building. So buildings never bring a valuable master plan to the building site where it might be damaged or lost. So instead, they prepare inexpensive, disposable copies of the master plan called blueprints. So similarly, the cell uses DNA's master plan to prepare RNA's blueprints. So DNA is the master plan, RNA is the blueprints. And the DNA molecule stays safely in the cell's nucleus while the RNA molecules go to the building protein sites in the cytoplasm, the ribosomes. So you can think of an RNA model as a disposable copy of a segment of DNA, a working copy of a single gene. And RNA has many functions, but RNA molecules are involved in protein synthesis only. So RNA controls the assembly of amino acids into proteins. Each type of RNA molecule specializes in a different aspect of this job. Okay, so the three main types of RNA are messenger RNA, ribosomal RNA, and transfer RNA. So, excuse me. Messenger RNA. So most genes can contain instructions for assembling amino acids into proteins. So the RNA molecules that carry copies of these instructions are known as messenger RNA, or you'll see the small letter M with the RNA in capital letters all hooked together. So they carry information from DNA to the partner cells. And proteins are assembled on ribosomes. These are the small organelles composed of 2 subunits. So the ribosome subunits are made up of several ribosomal RNA. And this is written as a small letter R and then the capital RNA. So these ribosome subunits are made up of several ribosomal RNA molecules and as many as 80 different proteins. Transfer RNA. When protein is built, a transfer RNA molecule transfers each amino acid to the ribosome as it's specified by the coded messages in messenger RNA. So the transfer RNA is written similarly. There's a small t and then a capital RNA. So how does a cell make RNA? In transcription, segments of DNA serve as templates to produce complementary RNA molecules. So most of the work of making RNA takes place during transcription. And during transcription, segments of DNA serve as templates to produce complementary RNA molecules. So the base sequences of the transcribed RNA complement the base sequences of the template DNA. In prokaryotes, RNA synthesis and protein synthesis take place in the cytoplasm. In eukaryotes, RNA is produced in the cell's nucleus and then moves to the cytoplasm to play a role in the production of proteins. Our focus will be on transcription in eukaryotic cells. You and I have eukaryotic cells. So transcription requires an enzyme known as RNA polymerase that is similar to DNA polymerase. And you can look at the pictures of this in the pages on the text. So RNA polymerase binds to the DNA during transcription and it separates the DNA strands. RNA polymerase then uses one strand of DNA as a template from which to assemble nucleotides into a complementary strand of RNA. So next let's talk about the promoters. So RNA polymerase binds only to promoters, regions of the DNA that have specific base sequences. So the promoters are signals in the DNA molecule that show RNA polymerase exactly where to go to begin making RNA. Similar signals in DNA cause transcription to stop when a new RNA molecule is completed. So, RNA editing. RNA molecules sometimes require bits and pieces to be cut out of them before they can go into action. The portions that are cut out and discarded are called introns. That's introns. In eukaryotes, introns are taken out of the pre-messenger RNA molecules while they are still in the nucleus. And the remaining pieces, known as exons, EXONS, are then spliced back together to form the final messenger RNA. Biologists don't have a complete answer to why cells use energy to make a large RNA molecule and then throw parts of the molecule away. But some pre-messenger RNA molecules may be cut and spliced in different ways in different tissues, making it possible for a single gene to produce several different forms of RNA. Introns and exons may also play a role in evolution, making it possible for very small changes in DNA sequences to have dramatic effects on how the genes affect cellular function. You guys have a good day.